Hello everybody, I'm Borja, and in this video I will present you my scientific poster on the investigation of the mechanisms behind sex differences in Alzheimer's disease using single-cell technology. Alzheimer's disease is a neurodegenerative pathology that affects millions of people worldwide. An interesting observation is that there are sex differences in the prevalence and clinical course of the disease. This raises the question of the underlying mechanisms related to biological and hormonal factors. This is where single-cell technology comes into play. Single-cell technology has emerged in the last decade as a valuable tool, generating a large amount of data and allowing a much more precise approach. This allows us to investigate cellular heterogeneity in diverse contexts, including neurodegenerative diseases such as Alzheimer's disease. This work aims to detect and characterize molecular and functional sex differences based on the meta-analysis of single-cell studies in the prefrontal cortex of Alzheimer's disease patients. To achieve this objective, the first step was to compile studies from public repositories focusing on Alzheimer's disease, which also take into account the sex of the patients. For each of these studies, an individual analysis has been carried out. Using differential expression analysis, we have identified the genes that are over- and underexpressed in each cell type for each study. In this way, we have obtained genes differentially expressed only in females, only in males, and genes whose expression varies according to sex. To make the results more robust, we have performed a meta-analysis to combine the results of multiple studies to obtain a list of common genes for each cell type. These common genes provide us with the key information to better understand Alzheimer's disease and the associated sex differences. In addition, we used functional enrichment techniques to analyze the common genes identified. This allowed us to detect enriched co-terms for each sex, which we grouped according to similarity of biological functions, giving us an idea of the key diseases-related processes for each cell type according to sex. Based on this process, the main biological functions detected for each cell type have been represented according to the sex of the patients. First, we have astrocytes, for which altered motility and cytoskeleton processes have been detected in men, while in women, metabolism, cell adhesions, the sodium-potassium pump, and the WNT signaling pathway were overexpressed. As for neurons, males, again, show alterations in the cytoskeleton and alterations in glucose and insulin metabolisms. Females, in this case, show modifications in chromatin and histone as a characteristic feature. For OPCs, we see an alteration in cell adhesion, this time in males, together with alterations in actin filaments and synapses. Females show alterations in telomeres and in the WNT pathway, again, as well as problems with lipoproteins. For microglia, we have altered functions typical of this cell group in men, such as the immune system or phagocytosis, as well as cell motility. However, for females, we have altered deregulated genes related to ubiquitination processes and cell vesicles. Finally, for oligodendrocytes, we have processes related to lipid and protein metabolism, in addition to the MTO-RC1 pathway for men, while in women, we have alterations in the JAK-ST pathway, calcium signaling processes, and genes involved in the accumulation of myeloid filaments, one of the main factors in the development of Alzheimer's disease. Preliminary results so far indicate the existence of sex differences in gene expression between different cell types in Alzheimer's disease. These findings support the importance of studying cellular heterogeneity and the mechanisms underlying sex differences in disease. These differences show increased cytoskeletal, immune system, and lipidemic processes in males, while females show alterations in neurotransmitter, catabolism, and gene expression regulatory processes. However, further analysis and in-depth functional enrichment is required to strengthen and extend the findings. I appreciate your attention and your time to hear about my research. If you have any questions or would like more information, please do not hesitate to contact me. Thank you.